soon as that comes on.
And then you can just open your...
Do you see anything? Okay.
I don't know why it's...
Do you know it's recording?
It's recording.
Okay. All right. I think we're recording. Good. Ready to go. Hi, I'm Dr. Terry Harrison, as Dr. Beaumont said earlier. Nice to see you all. This room is not really a very warm and friendly room, is it? It's not so easy to be interactive and to, it's not the best place for a class, but we have to cope. If you have questions and you're waving and I don't see you, go ahead and call out, ask me questions when you want to. It tends to feel a little more formal in here. We try to keep it a little more information exchange as much as we can. So please interrupt and ask questions, and I will be asking you questions. I hope, you'll respond so that we can kind of make it a little bit of a dialogue. Today, what I have for you is a very introductory lecture. You probably know all this material already, or a lot of it anyway. But just to make sure everybody's on the same page and you're all up to speed with it, I'm giving you an introduction on the organization of the central nervous system. There are several levels of organization, of course, to the central nervous system, so I'm just going to be talking very briefly about several different levels of organization and how we study and look at the particular level of organization on which you're going to be spending most of your time, which is the sort of systems level. But let's just go ahead. We're just going to sort of touch our toes into a little bit of anatomical terminology for the brain. Again, how many of you have had a neuroscience course before? Just raise your hand and let me. So there are a few of you out there. Don't want to claim it because you don't want to let anybody think you're an expert or anything, right? But you're going to be a little bit ahead of the game because at least you know some of the terms. There's a lot of nomenclature. And to make things worse, there's lots of places where there's several names for things and they get used, so life gets a little complicated. All right, so start globally. The nervous system has two main components, the peripheral nervous system, the central nervous system. The peripheral nervous system is basically composed of the peripheral nerves and various types of ganglia. The central nervous system is composed of the brain and the spinal cord. Of course, that's where you're going to spend most of your time here. But we're not going to completely forget about the peripheral nervous system. We know you've already had considerable experience with the peripheral nervous system. You've looked at nerves in anatomy. You follow them through the body. You know what a spinal nerve is. I think you know that the sensory part, this is the sensory part of the peripheral system, this is the motor part of the peripheral system. They're sort of separated conceptually here, but you know that they're intermeshed. The sensory side is composed of afferent nerve fibers. Afferent fibers are axons that approach the brain. They're the sensory fibers. They have their cell bodies in ganglia. For the lower part below the neck, they're in the dorsal root ganglia. Their axons proceed into the spinal cord or the brainstem. Let me give you a couple of terms right off the bat here. So I'm going to divide the brain into two sections for right now, two components. Uh-oh. Boy, this is really off. Sorry. I'm drawing over here, and I'm showing up over there. This is going to be hard. This is not working. Ew. Okay. That upper part of the brain that's inside that circle, that is the forebrain. This is not working. All right, guys, sorry about that. Can you spell forebrain for yourselves? Okay, that upper part of the brain is the forebrain. Between the spinal cord Just going to have to show you on there. The best laid plans are subject to technology. Between the spinal cord and the forebrain, which is this area up here, is the brain stem. Okay, so those two terms I'm going to be using when I talk about the rest of what we're talking about today. So the sensory fibers from the periphery come into the brain, in the brain stem, or into the spinal cord, the dorsal roots. I think that's something you all know. On the motor side, the efferent side, where axons exit the spinal cord or the brain stem, the cell bodies are located in the spinal cord or in the brain stem, and they send their axons out to the skeletal muscle, the smooth muscle, the cardiac muscle, et cetera. Now, on this side, there should be some ganglia also, right? The autonomic ganglia. They're not in there. There are a lot of things wrong with this diagram. I mean, these are supposed to represent neurons, little circles, soma. Axon is an arrow. These are supposed to be inside the spinal cord, right? Okay, the motor neurons are in the spinal cord, not on the outside. And they are, I put this one in myself, so it's actually inside the brainstem. I was talking about dorsal root ganglia before I got distracted by not being able to draw. The dorsal root ganglia, of course, are for the spinal nerves. There are sensory ganglia associated with the cranial nerves also, for the sensory input that comes in from the head. The nose and the eye are a little special. They don't have any ganglia associated with them, and they actually come into the forebrain. You'll learn all about that in the next few lectures. In a week or two, you'll get all the anatomical details about that. But so that's basically all I'm going to cover for the peripheral nervous system. You guys know it, and we don't spend a lot of time on it, but you are going to have to know some of it because we will have a unit where we're talking about neuropathy, plexopathy, and radiculopathy, or nerve root disease. And they're going to be saying things to you like, okay, so here's the peripheral symptoms. Okay, what nerve root are we talking about here? All right, so that's going to be part of what you're going to have to bring from your gross anatomy learning when we get to that point. It's a big part of neurology, dealing with those issues. Okay, so I'm sure you've all in gross anatomy got to see the brain and you got to see the spinal cord. You may not have seen something quite like this. This is a dorsal view of the brain and spinal cord. All you can really see here is the part of the forebrain that is the cerebral cortex from this view. And of course, this is the spinal cord with all of those spinal nerves attached to it. No biggie. Was there anything else that I wanted to say about this? We've got forebrain and brainstem and spinal cord. Okay, and I think that's got it. You're all clear about afferent and efferent? Yeah, good. Okay, so jumping from the global level of organization, I'm going right down into the cellular level, okay, and the cells of the brain and spinal cord. I'm sure you know already that the two characteristic cell types of the brain are the neurons and the glial cells. And these little diagrams here in the yellow or gold are sort of cartoon neurons, right? There are some neurons that actually kind of look like this in the brain, but not very many of them. Most of them are the motor neurons. Neurons have enormous variation in shape, and I will show you some of those things in a few minutes. Some of you have already seen some of that in histology and so forth, but it does illustrate the fact that those neurons are very polarized cells. Yes, this is very basic stuff. There's a soma, and apical, or emerging from that soma, are the dendrites and a solitary axon. The dendrites, of course, are the receptive end of the cell. There will be synapses coming into it from other neurons. That's the area where it gets its input, as well as on the soma. The axon is what conducts an action potential, a unidirectional activation of the system down to the terminals of the axon where it can then communicate with another neuron. So this is kind of a cartoon rendering you often see, this showing dendrite, soma, and cell body. And they're also here illustrated 5 of the 6 types of glia that I'm going to talk about today. The oligodendrocytes are a type of glial cell that generates the myelin sheaths that surround the axons in the central nervous system. They're sort of analogous to the Schwann cells in the peripheral system. However, in the central nervous system, an oligodendrocyte will send a process to many different neurons and only cover part of each neuron. So each neuron has contributions for many different oligodendrocytes to make the myelin coating that surrounds it. The microglia, you probably know, are the immune cells of the central nervous system. They act like the macrophages, really, of the central nervous system. That's really all I'm going to say about them. The astrocytes, or astroglia, same thing, 2 names, are very complexly associated with all of the cell types in the central nervous system. As it's illustrated here, they have many processes. They contact the blood cells, they contact the neurons, they contact each other, and they also contact the surface of the brain. Though it's not illustrated there, that's an important component. And here we have something called a polydendrocyte. There are actually 2 cell types now that have been identified that are basically precursor or stem cell forms that are present. The polydendrocyte is a precursor cell that can produce new oligodendrocytes. There's also a glial stem cell, which is illustrated down here. Oh, I need to get rid of that guy. Let me see. This is not good, Eric. It's really acting up. Okay. Glial stem cells can give rise to any of the cells in the brain, including new neurons. There's no, as far as I know yet, there's no definitive demonstration of this happening in the human brain, although we know it happens in mammalian brains in restricted places under restricted conditions. And we're going to talk more about that towards the end of the course, where we talk about rehabilitation, recovery from injury, and that kind of thing. But those cell types can give rise, the glial stem cells can give rise to any of the types of glia or neurons. This shows you in a little more anatomically correct versions of all those cell types that we just talked about. The astrocyte, with its enormous number of processes interacting with all the other cell types. An oligodendrocyte here with several processes, each of which is going to generate myelin around many different neurons. It's a microglial cell. Again, here it's called an oligodendrocyte precursor. It's got a slightly different name on the other side, but on the other slide, but basically it's the precursor for oligodendrocytes. And the glial stem cell is illustrated there. So here's some example of real neuronal morphologies. As I said, some of them look like those little gold neurons on that picture, with the sort of lots of little dendrites coming off of a cell body, and in a very kind of, what is the term for that? Pardon? Well, no, I'm thinking of the way there's a name for that particular morphology, and it's just escaping me right now. But they look like motor neurons. A lot of the motor neurons that will send axons out of the brain have that kind of morphology. But the neurons themselves in the rest of the brain have an enormous number of types of morphologies, and their morphology is very related to their function. So specific cells in specific locations doing specific things have very unique kinds of morphologies. We don't really bother you too much with most of them, but here's some examples and a couple of things that you will be learning about as we go on. I think if you've taken histology, you've seen an example of a retinal bipolar cell, right? It's a very simple neuron. It's got a single apical dendrite and a very single axon, very simple axon. It's not very branched. It just has a very small area on which it makes synapses on the next cell. Very small elaboration of the dendrite. This, on the other hand, is a Purkinje cell from the cerebellum. Here's the cell body. This is the dendritic tree in 2 dimensions. Okay, it's really a 3-dimensional cell, although it's not as big in the 3rd dimension as it is in this 2-dimensional area. But every single one of those little branches is going to have thousands of synapses on it. So it gets an enormous amount of contact from other cells. This is the axon. And here it's showing you that an axon can be complicated too. An axon can branch. We call these branches collaterals. They're collateral branches, if you will, but we just use the term collaterals for a major branch in an axon. One collateral can go one way, and the other collateral can go to a different target. Another type of cell, which I'm going to bring your attention to because we talk about this type of cell without really referring to it a lot, and this is the pyramidal cell. Pyramidal cells are the major output neurons from the cerebral cortex, from the hippocampus, from some of the other structures in the forebrain. If you're not sure what an output cell is, I'm going to cover that concept in a few minutes. So here you can see the cell body of the pyramidal cell. It's kind of triangular, which would be in 3 dimensions, kind of pyramidal, so that's part of the reason it has that name. It also has that name because of the dendritic structure of it. They all have these apical dendrites, which project from the cell body in the cortex up to the surface of the cortex. And they also have these basolateral dendrites, which extend around them in the same layer, basically, or near the layer where the cell body is. So that's kind of triangular too, or pyramidal in 3D. So you can see that also as a pyramidal cell in that regard. And again, a fairly complicated axon that doesn't just have one projection. It can go to several different places. When we talk about these in systems, we don't always talk about the collaterals. Occasionally we do, but we're talking mostly about the major output of those cells. And just to show you that there's really is a range, there are also within the central nervous system cells that really don't have any dendrites. They're like the pseudo-unipolar cells of the dorsal root ganglia. If you don't remember that, we'll go over that again in a few lectures from now. And there are some cells that have no axons. They just interact with other neurons through their dendrites. And that's not, neither of those is very common, but those things do occur and are important. And I put this here to remind myself to, of course, tell you that there are other cell types in the brain. The brain has got lots of blood supply, so every type of cell associated with blood vessels is going to be found in the brain. And the brain is lined with this single layer of cells, the ependymal cells, that lie between it and the fluid that surrounds it, the cerebral spinal fluid. So there's always the ependymal lining as well. Major cell types, a little bit about their morphologies. You're going to be hearing more about this, I think, in your next. Is that better? Okay. Of course, the synapse is a really important part of nervous system function. You've probably covered this, some of you at least. Have taken courses where you've heard about synapses before, so I'm just going to briefly talk about synapses. Most of the synapses in the brain are chemical synapses. The synapse is it's the spot where one neuron communicates with another neuron. Typically, an axon, right before it's going to make contact with its neuron target, is going to break up into a lot of little fine endings. And each one of those little fine endings is going to have a little swelling at the end of it, which is called the terminal bouton, or button is what it's called in this particular figure. The little terminal field of the axon is what is referred to as all these little branches and how they make contact with the postsynaptic cells is called the terminal field of the axon. Each bouton, of course, contains the synaptic machinery. The specialized areas of the axon are making contact with specialized areas of the postsynaptic cell. The changes in the voltage characteristics across that membrane when an action potential invades the terminal cause a release, influx of calcium, which causes, through various things I'm not going to go into, results in the release of a neurotransmitter that crosses a small gap, the synaptic cleft. There are specialized receptors on the other side which bind that chemical messenger. That binding, of course, institutes a whole lot of intracellular changes, which then can determine the response of the cell to that communication. You will hear a lot more about synapses and receptors and neurotransmitters, again, in another lecture or two coming up. There are also electrical synapses in the central nervous system. There aren't very many, but they're there and they're important. Electrical synapses are ones that are either based on gap junction channels or hemichannels that allow a direct electrical communication between the cytoplasm of two cells. So any electrical changes in this cell are directly communicated to this cell without the necessity of a chemical mediator. We're not really going to talk about them much in the course. I want you to know they're there, but that's about all we're ever going to say about them, I think, for the rest of the course, unless somebody brings them up in dealing with the thalamus, which is possible. But so the synapse, its basic structure. All this is pretty familiar to you, yes? I'm not teaching you anything particularly new at this point. So what I'm going to do now is just show a few pretty pictures. It's always fun to do. This is a stained hippocampal neuron in blue, showing the cytoplasm of the soma and the dendrites of an individual cell. And in red and green and yellow, because red and green put on top of each other come out looking yellow in this kind of figure, are all the synapses that are being made on that neuron. Okay? Dendrites are just lined with them. The soma also has a bunch of them on it. But there are hundreds and hundreds of those things, and that's just the proximal dendrites. Those are just the dendrites that are close to the cell body. Those dendrites can extend ways away, and they're full of synapses. And this is another one from a retinal neuron. In this case, the cell and the cytoplasm are in green, the dendrites are here, and everything is pretty much in yellow, showing the synapses that are on that particular cell. There aren't just a few synapses on cells, okay? There's tons of synapses on cells. And if you look at them carefully, you see in a lot of cases you can tell that this synapse seems to be, removed from that dendrite a little bit. Have you guys heard the term dendritic spines? Those synapses are out on dendritic spines, and if you just blow up a dendrite and just look at a dendrite itself, you can see that all along it there are these little special projections that stick up away from the dendrite. And typically, the axon will terminate on the little dendritic spine. Now, these cells aren't naked in between the spines. There isn't the synapse. There's the end plate or end foot of an astrocyte. Okay, same on the cell body and also on the axon. If it's not got myelin wrapped around it, there's going to be an astrocyte that's covering it. So there's no naked neurons in the nervous system. Okay, so now I'm going to use the words of Gerald Edelman here to just kind of sum up this part. Do you all know Gerald Edelman? Dr. Edelman was a Nobel laureate. He received the Nobel Prize for describing how the immune system can function to recognize the wide variety of antigens that it recognizes and for describing the structure of the antibody. And then he went on, after he did that, a few years later, he discovered cell adhesion molecules, a whole class of cell adhesion molecules and what they're good for. And then towards the end of his career, he decided to start doing some really tough work, and he started working in neuroscience. Now, as far as I know, he didn't have any totally revolutionary output for what he did with neuroscience, but he did sum up a few things that I think are interesting. So here's my gee whiz slide. A newborn baby's brain comprises a massive population of neurons, approximately 100 billion cells. And those that survive the initial phases of growth and development will make approximately 100 trillion connections with each other. A sample of brain tissue the size of a match head contains about a billion connections. And if we consider how these neuronal connections might be variously combined, by which he means how you could describe the different kinds of connections it could make, the number of possible permutation becomes hyper-astronomical, in the order of 10, followed by millions of zeros. So that kind of tells you the problem with trying to understand how the nervous system works. It's just too many of these things. The young brain contains many more neurons than will ultimately survive to maturity. And this redundant capacity is needed because neurons are the only cells in the body that cannot be renewed, and because only those cells and networks best adapted to their ultimate purpose will be selected as they organize into neuronal groups. Okay, so the second part, after the gee whiz part, the second part kind of alludes to a couple of things. One is to the fact that brains change with development as neural systems organize. We're not going to talk about that until kind of the end of the semester. We'll talk about development and experience and how that shapes how the brain gets connected and hooked up. And then it says the cells and networks best adapted will be selected as they organize into neuronal groups. That's going to take me to my next level of organization. These cells don't exist as a gemisch in the brain. They all are structured into specific patterns of interconnections, right? And that's what gives us hope for analyzing it, because it's not just generally random. It's systematic and organized. And at this level, I'm going to talk about neural circuits, neural circuitry, describing the patterns of interconnections between neurons. Okay? This is really the simplest circuit that could exist. One neuron communicating with another neuron. This has all of the structural features I've just talked about. It's got the presynaptic axonal terminals, and this with boutons on them in the terminal field of the presynaptic axon. It's got a neuron with dendrites that are in contact with these axons. And describing this neural circuit would involve telling how many contacts are made and where they're made on this postsynaptic neuron. It would involve knowing what the transmitter was. It would involve knowing what the receptors were. It would involve knowing what the effect on the cell was, whether it was excitatory or inhibitory, and how strong an effect that was in terms of influencing the behavior of this neuron. So describing the circuitry gets very complicated. Now that's for a fairly simple neuron. This is a real circuit drawn in cartoon from the brain. This is the neural circuitry in the cerebellum. And it involves many different cell types, and they're connected to each other in very specific patterns. So if you'll just bear with me, and I'll go through this a little bit. The cerebellum, which is this part up here, gets axons from several different locations. The sources which send axons into the cerebellum are called the input neurons. Okay? This input, when it comes into the cerebellum, doesn't all do the same thing. Some of it makes contact with one very specific set of cells, granule cells and Golgi cells. Others make contact with a different cell, Purkinje cell. The cells that are internal to the cerebellum contact each other in very specific ways, right? These cells have axons that come up and connect with these cells. These cells up here only connect here, but they can also connect with axons of these cells up there too. Granule cells and Golgi cells interact, but these don't interact with Purkinje cells, and so on. So the pattern of interconnections is systematic. It's repeated thousands and thousands of times throughout the cerebellum. Every time you see a Purkinje cell, it has a certain kind of pattern of connections. So everybody get that idea, right? All of these neurons could be considered to be either interneurons or local circuit neurons because their axons stay inside the cerebellum. Ultimately, one type of cell sends an axon out of the cerebellum. It's the output neuron, okay? And that is what is going to go out and influence some other structures in the central nervous system, depending on its connections. Most of the time, we don't really bother you too much with this level of organization, okay? A little bit we tell you about the cerebellum, you're going to encounter this again, maybe a basal ganglia, maybe a little bit about the cortex. But every single structure in the brain has an organization to it like this, with a complex set of interactions that are going on in it. And most of the time, we're just going to kind of bypass that completely, okay, and not worry you about it. What we're going to worry you mostly about is what's coming in and what's going out and where it's connected to, okay? Okay, so that's the level I'm talking about next. The neural system. A set of interconnected structures responsible for a specific function within the brain. And there are two major components of neural systems. There are the nuclei and the tracts. And just so nobody gets confused about this ever, a nucleus at this level of analysis, a nucleus is a bunch of neuronal cell bodies. Usually they're dendrites too, okay? A tract is a bunch of axons from those neurons, the axons of those neurons, if you will, that collect together and make contact with some other structure in the brain. They travel somewhere, okay? So they're really part of the same cells, right? The nucleus and the tract have got one cell in them, if you will. Parts of a neuron, different parts of the neuron. The nuclei, I'm going to show you some pictures, more pretty pictures, of just what nuclei can look like in the brain. Some are simple, some are complicated. But they're usually identifiable anatomically as independent structures. Tracts you can also identify. The problem with tracts, really, and this is kind of supposed to be a little funny, although you probably won't think it's so funny. I think when neuroscientists, over the course of a couple of centuries, were discovering all these different structures in the brain, they had a competition going on about naming and who could come up with the greatest names for things, okay? Because tracts, these collections of axons that are interconnecting things, can be called by all different, I mean, you can't just keep it simple and call something an optic tract and a cortical spinal tract. Some names are like that, and it makes sense. Okay, cortical spinal tract means the cell bodies are in the cortex, and the axons go to the spinal cord. corticospinal tract. But there are other names for tracts besides tract. We have fasciculi. It's the same thing. It's a collection of axons. It's often applied to a collection of axons in as part of the cerebral cortex that interconnect parts of the cerebral cortex on the same side. But you'll see the term fasciculus, meaning tract. You'll see the term lemniscus, meaning tract. You'll see the term commissure, meaning tract. It's a specific set of tracts, the ones that go across the midline, that interconnect different sides of the brain, are commissures. There's peduncles. Peduncles are really big tracts that are so big they're kind of structural and hold things together. Then there's bundles, okay? Axons are often referred to as fibers, and we talk about fiber bundles, axon bundles. Some of these things have bundle in their name. And then there are other things that are fiber bundles, that are tracts, that just have names, that have nothing indicating that they are. Internal capsule, corpus callosum, fornix, these are important collections of fibers that you're just going to have to learn the names of. They're actually some fiber tracts that have different names in different parts of the brain, though they're the same fiber tract. It gets confusing. So always remember that you're tracing an axon. It's part of a cell. It started in a nucleus, and it's going someplace else. And if you damage the axon, it's the same thing as damaging the cell body, right? You can damage a nucleus and get some kind of dysfunction, or you can damage the tract and get the same kind of dysfunction because it's the same cells that you're damaging. Okay, and then there's one other thing I want to bring up, which is this kind of structure called a reticulum. In the brain, there are some areas where you can't really identify clear-cut nuclei and tracts. There are neurons in there. And generally talk about the reticular formation coming through parts of the brainstem and stuff. Very loose aggregates of neurons that aren't really identifiable into structures and tracts. I'm just going to bring that up because you'll hear that name. We'll talk about it later. Okay, so here's just some more pretty pictures and illustrations of some of these things. This is a drawing of a section through the brainstem, okay? It's in this plane, so I don't know what you're used to calling it. Axial is a name for this plane. I've put the dorsal surface is always shown up, and the ventral surface is always shown down. And all of these little lines and circles and squiggles and stuff are the nuclei and the tracts that are present in the brain at this level of the brainstem, okay? So what I'm going to show you is a section, a histological section of this area of this section. Of course, increased in size, and boy, resolution is not great on the screen, but I think you can see that this area here kind of corresponds to a group of cell bodies. This is a Nissl stain. So all of the cell bodies are being shown here. You have some dark purple cell bodies that are here. And then this kind of group of them is distinct from this group here or from this area of the tissue over here or over here. And in fact, that is the hypoglossal nucleus in cross-section. It's three-dimensionally, it's very simple. It's a circle. In 3 dimensions, it's a sphere, sort of an elongated sphere, okay? And it contains the motor neurons whose axons leave, bundle together and leave. They're actually running here. You can't see it very well, I don't think. But they run here. They exit the brain as the 12th cranial nerve, hypoplossal nerve, okay? So that's a very simple brainstem nucleus. This is another kind of nuclear organization that you often see. In this case, we're not as, we don't have as high a magnification, okay? We're looking at it less magnified. Again, we've got cell bodies stained. This is the olfactory bulb, and you can see that it's organized into layers. There's a layer of cells here, there's a layer of cells here, there's another kind of looser layer of cells up here. Layering is pretty common for a lot of the structures that you'll see, and we'll talk about many layered structures. A lot of them are sensory. The cortex is layered. Now, there are other kinds of structures too. So we're back to the same nucleus the same section we were a little while ago. I'm going to show you this area where this ridiculous looking squiggly thing is. Okay, and it looks just like that when you look at it in a section. Now this is a section that's been stained differently. Okay, it's been stained with a myelin stain so that all the fibers are black. And now where all the cell bodies are not stained, so they're clear. So this is a collection of cell bodies that exists in the brainstem, clearly identifiable. It has a ridiculous name to match its ridiculous structure. It's called the superior olive, okay? And again, it's just something really identifiable that you could pick out whenever you look at the section. And this is what your chore is kind of going to be, is to look at sections and learn to recognize these anatomically distinct components that constitute nuclei of the brain. This is another section. Now, this is a section of the forebrain, okay? And it's got on it part of the cerebellum. You see it's cut off. Again, we've pulled back out again. This is much lower magnification to get the whole brain stem with part of the whole forebrain with part of the cerebellum attached to it there. By the end of this course, you'll be able to recognize every structure that's present in that section, okay? It's got fiber tracts and it's got nuclei. And again, myelin stain, the fiber tracts are dark. There's a big one there. There's a big one there. There's one there running through the brain. There's a little one there. This is a collection of axons. This is a collection of cell bodies. Okay. This is another structure. And the reason I'm sort of showing you this slide is because we consider this is one structure. But it has areas that look, in terms of the staining, they look different from each other. Some of the organization, some of these structures, these nuclei, are organized into subgroups. In some cases, we're going to have to talk about recognizing subgroups based on their histological profiles. Okay, now, another way we're going to look at the brain is we're going to look at it in unstained sections, okay? This is a horizontal section cut this way, again, or you can call it axial. When you make a section like that, anterior is forward, is up, posterior is down. And you can see, even when it's not stained, that there is some contrast in there that helps you recognize different structures. Yeah? There's some very light areas, and there's some areas that are kind of gray, browny looking, slightly different variations in color, because this is, of course, fixed tissue, and the fixation is a little bit different in different parts. But we make the distinction between the white matter. Remember I don't know, they probably always tell you in gross anatomy that when you look at a nerve in your cadavers, it looks kind of white. But if it was in a real person, it's really shiny, right? Because the myelin is this white, shiny stuff. So it's sort of a glistening white. This is all myelin, white matter, okay? So this is all fibers. All this light stuff is fibers. This darker looking stuff are the areas of the cell bodies and dendrites. So that's the nuclei. So here you can recognize there's a nucleus here. It's separated by white matter. from other nuclei. Here's one here. Here's another one here. And then there's all of this, the cell bodies of the cerebral cortex in that mantle that lines the surface of the brain. Okay, so you will be looking at some of these also and learning to recognize the nuclear structures that are present. You know, why don't we take a break? I have a little more after this for you guys, and we'll just start again here.
I think it was.
Okay, let's resume. Maybe we can get out of here kind of early. I haven't got that terribly much left to talk about. So let's just make some of this a little more formal. I've already started to talk to you a little bit about making sections and calling things in this plane and that plane and so forth, different things. So let's just formalize that. Before I start, my course director said that sometimes my voice gets too low. If you guys start having trouble hearing me, please let me know, okay? I've been told before I don't project that well, so just wave at me or something, okay? When we want to look at the brain, we want to examine its organization. We have to do, for several centuries, up until the end of the last one, we had to fix the brain to be able to section it and look at it. The brain is really gooey. If you try to take the brain out of the skull, without it being fixed, it's like not very well set Jell-O. You try to put your fingers under it and lift it up, and it will kind of drip out between your fingers and stuff, okay? It's really very, very, very soft. So not a whole lot could be learned by just examining it. When they finally developed ways of fixing it so that it was hard, it could be taken out, it could be sectioned, it could be looked at. That's when people started learning things. And they systematized the way this was done, of course. Orientation is important, and identifying the orientation of a section you're looking at is going to be the first thing you got to do in order to make sense out of it. This just shows you the three planes in which sections are usually made of the forebrain, okay? And there are a couple of different names for them. Again, as I said, this seems to be always a couple of different names. For sections made in this plane, which you might call a cross-section, it's usually called either a coronal plane or a transverse or frontal plane, going this way, like this, or like this one here. This is the lateral view of the brain. And this dimension, this direction, this plane of section is usually referred to horizontal. Horizontal was the sort of original anatomist's name, right? With imaging and things, people generally refer to this now as axial. You'll see both horizontal in atlases and things like that, or axial when you're talking about imaging. And then there's this lateral plane, from lateral to mid, is called the sagittal plane. I think that's the only name I know for that one. Okay. Now, with the brain, with the nervous system, things get a little more complicated. Oh, what do you know, another illustration. This didn't project as well as I'd hoped, but these are MRIs in the three different planes. Here you have a coronal or transverse scan of the front of the brain. The ears are over here. This is a horizontal or axial image. Okay, the eyes would be up here. We're above the level of the eyes, apparently, so you don't really see them. That's, again, that's the front, anterior and posterior. It's usually, clinically speaking, it's conventional to show this with the person's left side on the right. I think you've heard this before, right? The patient lying down, you're looking up through their feet. So you're seeing, from your perspective, on your right is their left side of the brain. And then a sagittal section. This one is a mid-sagittal section right along the midline between the two hemispheres of the brain. And you can see the nose where the eye would be here. You're going to be seeing a lot more of those. Now, the problem is that we have not just the forebrain, but we've got the brainstem. And we stand on 2 feet. And we're weird, right? This, I think, this is ridiculous. They say, in the worm. Yes, we all know worms have cortexes, right? And spinal cords. No. Maybe in the cat. 4-feeted, you know, 4-legged animals. Everything is in one plane as far as the neuraxis is concerned, the central nervous system, the axis of the central nervous system. Anterior and rostral are the same, right? The nose is in the front, that's anterior. And caudal and posterior are the same. The tail is at the back end. Ventral and inferior are the same, and dorsal and superior are the same, right? But on 2 feet, you end up with a neuraxis that takes a right-hand turn, okay? And things get a little more complicated. Anterior and ventral are the same for the spinal cord and the brainstem, but then you make a right-hand turn, and suddenly ventral and inferior are the same, okay? Dorsal and posterior are the same until you make the right-hand turn, and in the forebrain, superior and dorsal are the same, okay? And caudal is, you know, to the tail, and rostral is to the nose. And that's anterior up here, but this isn't really posterior, right? So it gets complicated just because of this bend. And you're going to have to be a little flexible about thinking about that. When we show you histological sections of the brain, which you'll look at to learn where various structures are in relation to each other, as I showed you before, we put dorsal up and ventral down. Now, at this level of the brainstem, anterior and ventral are the same, and posterior and dorsal are the same. When we do a scan, if we did a horizontal section or an MRI scan, and we put that image up, anterior is up, posterior is up, and look at where the brainstem is. It's upside down compared to this one. Uh-oh. Okay? So you got to think in 3D a little bit when you're dealing with this. Okay? Because you're going to learn how things are oriented to each other in this kind of a section. But then you're going to see images like this. You've got to remember that this is this part. Right? Luckily, you can, the shape tells you. But you're going to not have to be surprised about this. You're going to have to expect what's coming. Okay. Now, I've introduced the idea of imaging, and I'm just going to mention different kinds of imaging that you might be coming across in specimens in this course. CT scans, computerized tomography. Okay, very common, very important clinically to do a CT scan sometimes. They're based on X-rays, basically. So you can't use them willy-nilly. You can't expose people to these things constantly, right? It's invasive and you can't give them too many doses. But they show you a lot of things. Here's a normal CT scan in a horizontal plane, right, of the skull. The bone is very dense, comes out white. The fluid that surrounds the brain and is contained in the internal ventricles of the brain is dark. And the tissue is sort of variations of gray. The white matter, the dense white matter, is a little bit darker. I don't know if you can tell that there. That kind of discrimination is a little hard to make in a CT scan because the resolution isn't so great. But you sure as heck can tell when there's been a bleed into the brain and it's coagulated, right? It becomes hyperdense compared to what the tissue ought to look like. This area here that looks normal is isodense with the normal. Okay, isodense just means it's the same. It's normal. In this particular scan, there's an area that's hypodense. It looks like it's just got fluid in it when it ought to have tissue in it. That's an area of cell loss, let's say following an infarct where the neurons have died in that region. You get that kind of blank. So they're very useful to show you damage that's occurring or has occurred and where it's located. CT scans do a good job. These are MRI scans. They tend to have, they're usually better, they're can be very high resolution. These particularly didn't scan very well and they don't look so high resolution to you right now, but it's one of its major advantages. It's much more high resolution and you can adjust the resolution. It's also not as invasive as we'll talk about in a little bit because you can use contrast media that you inject into the bloodstream to try to increase the contrast or change the contrast in the tissues. But you don't have to, and it's really based on something that doesn't require any invasive procedure for the patient. But one of the things that's very flexible about it is you can do it in any plane. If you have an MRI scanner, you can scan in any of the planes. And you can adjust contrast so that you can get sort of negative images depending on what you want to look at. This is called a T1 weighted MRI. And in these, the areas of fluid are like in the CT scan. The areas of fluid are dark and the areas of tissue are light. This is called a T2 weighted, and things have been adjusted so that it's a negative image. The areas of fluid are white. The area of tissue is dark. And when you do that, you actually can see a little more of the blood supply, the blood vessels that are running in the brain. So you can manipulate contrast in an MRI to look at specific aspects of the tissue that you're interested in. And then there's functional brain imaging. I'm sure you've all heard of this. You can certainly look at, well, those are all static images, essentially, right? You're just looking at the structure of the brain and something in it. These are ways of looking at actual neural function in the brain as it's ongoing. So you can look at neural activity over time when someone is doing something, and you can look at what goes on in the brain while that's being done. And that's been sort of revolutionary in our understanding of how some things work. The top two types are PET or SPECT, positron emission tomography, and single photon emission computerized tomography. These all rely on having some kind of radioactive element in being injected and taken up in the brain. And usually it's something related to metabolism, because the active neurons, ones that are going to be active when you're looking at something, are going to need more oxygen and more glucose and, more stuff. And so if you have radioactive glucose, they're going to be the ones that take up the most radioactive glucose, so you get a bigger signal from there. Okay? But the functional MRI is the one you may see in this course when we lecture about things, because this is the preferred way to do things if you can. It's, again, the least invasive. It's purely based on the native oxygenation levels in the brain. So if an area is active, it's going to be using a little more blood. It's going to be taking up a little more oxygen. So relative to other parts of the brain, the oxygen level is different, isn't it? And it's changes in local blood oxygen levels that form the basis for the functional MRI, and it's very non-invasive, so you can do as much as you want, basically, as your patient will tolerate. Many people are really claustrophobic in MRI machines. If you haven't run across that before, it can be difficult for some people. And when you get an image like that, this is sort of what you're going to get. This is combined MRI and fMRI, a static MRI image with a functional MRI image. When you do your computerized slicing through the brain, as you step down, you get an image every so often going down through the brain. You can layer on top of each other a static MRI and some measure of the activity of the neuron. The computer will assign some kind of color, based on what you tell it, to particular levels of activity of the neurons. So clearly in this series here, we have ongoing neural activity in these two parts of the cortex, and we also have a tumor indicated by the dense white matter. And you can project these things into a 3D realm as well, so that you can get a complete view. In this case, remember now, this is the person's left, and this is the person's right. In this case, the person was asked to move their left arm. And just in case this basic fact isn't like foremost in your brain, get it there. If I'm going to move my right arm, the left side of my brain is going to be active. If I'm going to move my left arm, the right side of my brain is going to be active, right? So this person was moving their left arm, the right side of their brain is active, okay? And as I said, you can get these kind of 3D images that you can, get a real sense in space of areas that are involved. Okay, so kind of end of pretty pictures and the beginning of our dipping our toes into more anatomical organization things, okay? I started out saying we have a brain and a spinal cord, and then I divided the brain into the forebrain and the brainstem. And guess what? We're just going to start making branches in this tree of subdivisions of the brain, okay? A lot of it is based on embryological origins. We start with the forebrain, which I can't draw on here because there's some kind of a spatial mismatch. I'm sorry. I'm just going to have to, I was hoping I could do that. I'm just going to have to run back and forth. Okay. So I told you in that first picture that the forebrain was this area up here, right? Most of the brain. It's mostly cerebral cortex, but it's got a few other really important things in it. It's actually composed of two parts. It's composed of the telencephalon, which is, again, hugely cerebral cortex, and includes some nuclei that lie below the cortex itself, but embryologically originate from the same source as the cerebral cortex. And the diencephalon. Now, the diencephalon, this is, again, a mid-sagittal section, right down the middle here, just like this one. So you're looking at the medial surface. You can see the diencephalon in here, and it looks kind of small, right, compared to that huge cortex. It contains two components, the thalamus and the hypothalamus. We look at it big, okay, you can sort of see that there's a region up here that's a little bit separate and different from this region down here, okay? That's the thalamus, that's the hypothalamus. That constitute the major components of the diencephalon. So we have telencephalon and diencephalon composing the forebrain. And then there's the brainstem. The brainstem, okay, so I took that picture and I just blew it up and showed you a photograph of a real brain over here. And a diagram about the same size. Looking at it, that's mid-sagittal. This is the lateral view of the intact brainstem. The brainstem can be divided into three areas. And just looking at it, I think they're fairly clear, right? Structurally. You have this basically kind of tubular part that's called the medulla, or the medulla oblongata is its full official name. It has some features on it that help you recognize it. It's got specific cranial nerves that come out of it at specific levels, right? But it's distinctly different structurally from this kind of swollen area that's right above it. That's called the pons. And above that is another area that again looks functionally different, structurally different, which is the midbrain. Okay, this one has cranial nerves coming out of it too that are characteristic of the pons. We've cut off this other big structure, very big structure called the cerebellum, so that you could see it without the cerebellum obscuring it. If I label the same areas on that mid-sagittal section, so again, now you're sort of looking internally at the internal surface on the midline. This kind of nondescript looking area, tubular area here, is the medulla. That fat, swollen, sometimes referred to as the belly of the pons area here, both of these are in the pons. Okay? And up above the pons is the midbrain. You can see the midbrain is kind of separated from itself here along the midline. This would be the area where the bumps are back here. These little hills, these colliculi is the Latin name for a hill, up here, and the rest of the midbrain down here. Okay? So, brainstem is made up of midbrain, pons, and medulla. Now, some of you, I think that's the next thing here, might be wondering if there's a midbrain and a forebrain. Isn't there a hindbrain or a backbrain or something? Well, there is a hindbrain. The hindbrain is everything in the brainstem that isn't the midbrain. Okay? So unfortunately, we have some different but overlapping terminology there. We don't use the term hindbrain very much. We refer to the brainstem or midbrain pons medulla. Okay. This, sitting on top of the midbrain, is kind of a three-dimensional view now of the diencephalon that I could only show you the middle surface of before. But this is the diencephalon sitting on top of midbrain. We're almost done. But then there's this picture. It sort of shows how this all nests. Okay? More terms. All right, but it's a nice picture because it shows you all in different colors what all the different names are, right? And how they all relate to each other. So we have the brain and the spinal cord. Okay? Spinal cord's no problem. Spinal cord. We have the forebrain, which I showed you before, right? This part. And on this, unfortunately, because they wanted to make it symmetrical, they didn't put brain stem in here. The brain stem would be the midbrain, the pons, and the medulla. So the brain stem is this. Embryologically, the forebrain contains the telencephalon and the diencephalon. The brain stem includes the mesencephalon, which is the same as the midbrain, the metencephalon, which is the cerebellum and pons. They originate from the same source embryologically. They're just different structures, but they're very related to each other functionally. And the myelencephalon, which is also the medulla oblongata. Okay. We use the term diencephalon a lot. We use the term telencephalon sometimes. We don't use these other terms generally, but you need to know them because it's embryology. It's part of what you're expected to know. Although other faculty will probably not talk about them very much. Was I going to say anything else? Oh, yes. I'm sorry. I was going to continue to take this out up here. So the telencephalon contains the cerebral cortex, and as I said, some deep nuclear groups. Among those nuclear groups are the basal ganglia, of which there are several. They are very involved in motor control systems, as well as other things, but mostly we focus on their motor function. Parts of the limbic system. If you haven't heard that term before, the limbic system is kind of a name that's hard to explain. It's mostly parts of the brain that seem to be regulating primarily affective, emotional, motivational kinds of behaviors and functions. Okay? There are a couple of nuclei in the forebrain that are associated with that function. And again, the diencephalon, the major structures there are the thalamus and the hypothalamus. So if you guys can get those under your belt, starting now, okay? When we get into the lab, we're going to be going deeper into those areas. We'll show you the basal ganglia and the 3 nuclei that make them up. We'll show you the limbic system nuclei and what their names are. Okay? We'll show you the surface of the telencephalon. We'll show you the names of the different gyri and sulci on the surface of the cortex, where areas are that do specific functions. Okay? We'll start to take you through some sections in these other parts of the brain, so you can begin to recognize what's characteristic of those levels and how you know what level you're in when you see it. Okay, so that's kind of coming up in your lab this coming week. There is posted on D2L a list of structures and terms that you are going to be experiencing in the lab and in lectures. And you're going to start learning where they are, okay? If you can read those before you go into lab, you'll know what to listen for and to look for as you go through a couple of different stations and people start demonstrating to you where these things are. Okay? So the next week or so, you're going to be doing two different things. You've got a lecture on, two lectures, one on cellular properties of neurons, and then on for neuropharmacology, essentially. Communication between neurons based on chemical synapses and their receptors for neurotransmitters, right? So you're going to be working on the cellular level in your lectures. And in the lab, we're going to be doing basically some gross anatomy, surface anatomy, and some gross deep anatomy of the brain at the same time. Okay? So get ready for it. Then when we come back, after those sets of lectures, I guess it's before the next lab, I will come back. And we will really kind of jump into some detailed neuroanatomy, starting in the brainstem. And dealing with the cranial nerves, how the cranial nerves give you a kind of an understanding where their nuclei are, give you a framework in which to understand the organization of the brainstem. From there, you go into all the sensory systems, okay? So we're hoping by the time you get into the sensory systems, you will have heard the words that we're going to use to describe the nuclei that are involved in the sensory system, because you will have had those in your lab, okay? That's kind of where we're going in the next, through this block. Okay, does anybody have any questions? Everything going okay here so far? Okay. You have my phone number, you have my email. I'm happy to see anybody, anytime. Just please, you're not necessarily going to find me if you just drop in. If you can let me know you're coming or when you might want to come, that works really well. And good luck with everything. Oh, can I just do one more thing for you guys before you leave? I mean, if you don't want to stay for this, it's okay. I was going to demonstrate for you the online atlas that you have that you can get through the Sylvius 4. It will take me just a minute to, I can't close this thing.